Dear students, welcome to EPG Pathshala. I am Dr. Imtiaz Hassan from Jamia Millia Islamia. Today I am going to talk on dynamic light scattering under the paper 10 of the Biophysics. In this module we will be discussing on the major components of the dynamic light scattering techniques. The objective of this module is to provide the detailed information on the basic concept of the dynamic light scattering techniques. Then we will discuss about the Brownian motion and the autocorrelation function. After that, we will discuss about the application of dynamic light scattering, then its advantages and limitations. After that, we will compare the dynamic light scattering with that of static light scattering, and at the end we will going to summarize the dynamic light scattering techniques. So now we will discuss about the dynamic light scattering. The dynamic light scattering, that's DLS, it measures variation in the scatter intensity with time at a fixed scattering angle, that is typically 90 degree, while the static light scattering measures the scatter intensity as a function of the angle. The DLS is used to measure the size of the particle typically in the submicron region, that's also referred as photon correlation spectroscopy or quasi-elastic light scattering technique. When particle is suspended within the liquid medium, it undergoes Brownian motion, and the larger the particle, slower the Brownian motion. So DLS actually monitors the Brownian motion with the light scattering. The speed at which the particles are diffusing due to Brownian motion is measured by recording the rate at which the intensity of a scattered light fluctuates. And the best thing is that smaller particles cause the intensity to fluctuate more rapidly than the larger particles, and thus DLS is also used to measuring the zeta potential of the particle and measuring or estimating the molecular weight of the organic compounds. The dynamic light scattering instruments, it actually use the light that and scattered data on the microsecond time scale. It can be used to determine the particle's molecular weight, size of the distribution, relaxation of the complex fluids. And now we will talk about the random motion of the particle, that is the Brownian motion, which leads to the random fluctuation in signal due to changing constructive and destructive interference of the scattered light. So the Brownian motion is actually occurred when particles or emulsion or molecules in suspension. In the Brownian motion induced by the bombardment of solvent with each other and themselves moving due to of the thermal energy. Temperature and the viscosity is therefore must be known for the calculation of the measurement of the DLS data. The velocity of the Brownian motion is defined by a property known as translational diffusion coefficient, that's usually given in the form of symbol D, and this process is the random, related to the size, of course related to the viscosity of the medium, and temperature is one of the important factor which regulates the motion of the particle under the Brownian motion. Now I will give you the mathematical description of the Brownian motion. The Brownian motion is simply derived in terms of the equation given by two well-known scientists, that's the Stokes and Einstein. The decay constant is related by Brownian motion to diffusivity by tau equals to minus dq square, where the q equals to 4 pi n by lambda sin theta by 2, where and the q square reflecting the distance of the particle that travels, and it fitted into the Stokes-Einstein equation, in which the diffusion coefficient D equals to k T divided by 6 pi mu r. The numerator k T represents thermodynamic properties, and the denominator represents the hydrodynamic properties, in which dH is the hydrodynamic diameter, d is the diffusion coefficient, k is the Boltzmann constant, T is the absolute temperature, and eta is the viscosity. Here we are going to show you that the basic component or basic principle of the dynamic light scattering. You can see here that if particles are in a Brownian motion, once the light comes to this, it is scattered in all the direction, and the scattered intensity is actually calculated or measured in terms of the time dependent manner, that is given as the intensity autocorrelation function. Here we will discuss about the instrumentation of DLS. The DLS is a very simple technique where one light source is required, then one is the sample holder where the particle or the sample is kept in the cuvette, and then one detector is required which is fixed at the right angle to the direction of the light. So, the dynamic light scattering is the fast technique used to study the dynamics in soft condensed matter. This method involves performing online autocorrelation of the intensity fluctuations of the light scattered from the sample. To obtain reproducible results from the DLS experiment, it is necessary to have source and detector free of self correlations. It also have capability of high speed signal to noise processing to perform online autocorrelation and means for maintaining the steady sample temperature. A digital autocorrelator calculates the discrete time autocorrelation of the variable with the time v with a decay time xt. You can see here how the component of DLS are arranged. You can see here the light source is the helium and neon laser which pass through a slit and then comes to the particle, and then it is the signal is collected by the digital controller, and of course the whole temperature of the whole system is maintained by the temperature controller. The mathematical calculation of the theta dependency is shown here, and this animation show you how the DLS performs its function. Here I want to discuss about the intensity autocorrelation, how the autocorrelation is derived. You can see here in the figure that scatterers are diffusing under Brownian motion with mean square displacement given by r equal to r square equals to 6 dt c. That is the Einstein equation. The correlation time c is time Tc is measured of the time needed to diffuse the characteristic distance in solution. This distance is defined by the wavelength of the light, the scattering angle, and the optical properties of the solvent ranges from 40 to 400 nanometer in a typical system. Values of Tc can be ranges from 0.1 millisecond especially for the small proteins to a days, that is for the glass and gels. The autocorrelation function is further described as the rate of change in scattering intensity by comparing the intensity at time t to the intensity at later time, that is t plus tau, and providing a quantitative measurement of the flickering of the light. Mathematically, the correlation function is written as an integral over the product of intensities at some time and with the some delay time, that is tau, that is mathematically expressed at g2 tau equals to 1 by t and integration from t0 t to 0 I t into I t plus t dt. Here the different times are shown in the graphical scale, which can be visualized by taking the intensity at t time and the intensity at t plus tau times followed by the same product, that is the t plus t prime and so on. That is the importance of autocorrelation function in the dynamic light scattering. Here I try to show you the chromatogram analysis or correlogram analysis. So, how the correlation function, autocorrelation function related to the size of the particle. You can see here for a larger particle, signal will be changed slowly and the correlation will persist for the long time. However, for the smaller particles are rapidly moving and the correlation will disappear quickly. You can see here for the very small particle, the medium range polydispersity, no large particles aggregates present at the baseline. So, in this slide you can find different types of particle having the different types of correlation functions. Now we will discuss about the diffusion, that is the major factor in the DLS. So, whatever we learn from the correlation time, knowing the characteristic distance and the correlation time, we can find the diffusion coefficient D. According to Stokes-Einstein equation, the diffusion coefficient is given as Kb into T divided by 6 pi eta Rv, where R is the radius of the equivalent hydrodynamic sphere and eta is the viscosity of the medium. So, if eta is known, we can find R, or if R is known, we can find eta. Let us talk about the protein hydrodynamic size, because the size of the protein is measured in terms of hydrodynamic radii. The hydrodynamic diameter of non-spherical particle is the diameter of a sphere that has the same translational diffusion speed as the particle. If the shape of the particle changes in a way that it affects the diffusion speed, then the hydrodynamic size will change. For example, a small change in the length of the rod-shaped particle will directly affect the size of the particle. Now, we will discuss about the conformation of the proteins also directly related to the size of the particle. So, the conformation of the protein usually dependent on the exact nature of dispersing medium. As conformation changes will occur, it usually affects the diffusion speed. We know there are some factors which influence the protein hydrodynamic size are the molecular weight of the molecule, the shape of the conformation of the molecule, and also whether the protein is in native or the folded state. Generally, proteins are in a spherical shape, but due to conformational changes and also aggregation from it forms aggregates of the spherical light objects. The another important factor in the DLS technique is the polydispersity. In the light scattering areas, the term polydispersity derived from the polydispersity index, a parameter calculated from the cumulant analysis of the DLS measured intensity autocorrelation function. In the cumulant analysis, a single particle size is assumed and have a single exponential fit applied to the autocorrelation function. The polydispersity can be cut into three areas. A sample is monodisperse if the polydispersity is less than 20%. It is a medium disperse if the polydispersity lies between 20 to 30%. And it is known as polydisperse if the polydispersity value is more than 30%. If one were to assume a single size population following a Gaussian distribution, then the polydispersity index would be related to the standard deviation of the hypothetical Gaussian distribution in a fashion that is expressed as polydispersity equals to sigma square divided by z square d, where z d is the z average value of the intensity weighed means hydrodynamic size, that is the cumulant mean of the ensemble collection of the particle. Now, we will talk about the obtaining size information from the correlation function. A fit of a single exponential to the correlation function, that is cumulant fit, to obtain the average hydrodynamic radii RH is equally Z average diameter, and the estimate of the width of the distribution, that is the polydispersity index, or fit of a multiple exponential to the correlation function to obtain with the distribution of particle size. The size distribution obtained is a plot for the relative intensity of the light scattered by particle in various size classes and is therefore known as intensity size distribution. If the distribution by intensity is a single or fairly smooth peak, then there is a little point in doing the conversion to the volume distribution using the Mie theory. Here we will discuss about estimation of particle size by using high dynamic light scattering techniques. Let us have an example of two particles which have one of having size of 80 nanometer and other have the size of 8 nanometer. This particle will scatter at 10 to the power 6, this 1 million times more than the particle of 8 nanometer. You just assume that the size is of 10 times, but the scattering intensity is 10 to the power 6 times. The distribution of the total light scattered by the small particle will be extremely small, and thus DLS gives the diameter of the sphere that moves or diffuse in the same way as your sample. You can see here the intensity of the two particle that's represented in the hydrodynamic plot. So, you can see that the population or relative population of the two particles are in a range. By taking the my theory, it is possible to convert intensity distribution to the volume by taking the simple equation V equals to 4 by 3 pi r cube, where r is the d by 2 and 4 pi d by 2 cube equals to 4 pi d cube by 8. So, volume is directly proportional to d to the power cube, however, the number is directly proportional to the d. So, you can see here, if it talks about the intensity and the volume, you find the in terms of intensity it is the ratio is 10 to the power 6, but if talk about the volume, the ratio is 10 to the power 3, while the numbers are equal because both are in the same number. Now, we will discuss about the application of dynamic light scattering in biology especially. So, you can see here the protein aggregation phenomena or the formation of bigger molecules from the smaller molecules can easily be detected by the dynamic light scattering. Not only the formation of particles, the kinetics can also be measured. You can see here it is used to study the monomer size and shape, it used to estimate the polymerization kinetics, it used to study the gel network structure is formed and interaction within the molecule also. So, the simply DLS is used to estimate what factors cause to promote the aggregation, how proteins can be protected from the aggregating and what are the kinetics. These things can be estimated by implying the DLS technique. You can see here in a graph, if we take the log 10 of the hydrodynamic radii versus the log 10 of the light scattering intensity, you find a straight line can be formed which used to estimate the different types of aggregates formed in the protein, and that can estimated by the dynamic light scattering techniques. Here we discuss the implication of DLS in the measurement of thermal stability of the protein. The thermal stability can be measured by the measuring size and scattering intensity as a function of temperature. You can see here in a graph the protein melts under the influence of heat when the molecule denature, leading to massive aggregation. This transition is visible in light scattering studies as a significant increase in both size and scattering intensity. The marked point where both the size and the intensity start to increase significantly is called melting temperature. Here you can see the 56 degrees Celsius is a melting temperature. This melting point is a representative of stability of the protein under the current solution condition. The protein that has a larger melting point is more stable. So, the DLS can be extensively used to study the stability of proteins in terms of its thermal stability. Here we will discuss the dynamic Debye plot. If there is a concentration dependence in the results is obtained, a dynamic Debye plot can be constructed. It is recommended that if we are making DLS measurement on any sample, we should check that the results we get are independent of concentration. We make the measurements of the diffusion coefficient at several concentration of the sample, and we plot the diffusion coefficient in the dependence of concentration and then extrapolate to 0 concentration to determine the diffusion coefficient in the line of infinite solutions, and it is expressed mathematically here, and a plot is shown here that is known as dynamic Debye plot to make your data concentration free or concentration independent. The DLS technique is also used in the nanotechnology to measure the size of the nanoparticle. Here you can see here there are two types of particle. One are the coarse particle that give what kind of intensity you can get in the coarse particle, and for the fine particle. For DLS analysis, the hydrodynamic diameter is measured through optical detection of the Brownian molecular motion of the particles in a liquid. The thermally agitated liquid molecule collide with the particles, causing a random movement or diffusion. According to Stokes-Einstein equation, the diffusion velocity are inversely proportional to the size of the particle, which can be used to estimate the nanoparticle size and mostly used in the area of nanotechnology. Now we will discuss about the application of DLS in the protein crystallography. Dynamic light scattering is an established technique in the field of protein crystallography. It is used to measure the hydrodynamic size, polydispersity, and aggregation effect of the protein samples. These are the important parameters for the crystallization of proteins. DLS is also used in the area of nanotechnology research for the accurate and fast size measurement of the nanoparticles, and made up of made from the different materials. Now we will talk about the correlation function of lysozyme sample and the calculated distribution of the hydrodynamic radii. The main peak you can see that obtained at 93.8% of the scattered light is found, that is corresponding to a hydrodynamic radius of 1.95 nanometer, and the polydispersity is of the sample is 19%. The lysozyme sample shown here is therefore monodisperse and no protein aggregates are present. Such a DLS results indicate a good chance of crystal growth, but it is not a guarantee. So the DLS is widely used to check the protein sample that whether it is crystallizable or not. The other application of DLS are to estimate the population of aggregate, especially large and small aggregates for small molecules, proteins or lysozyme. We can check the quality of biomolecules prior to running the costly analysis such as SPR or SANS. It used to analyze thermal stability and chemical denaturation, differentiating pure unfolding from the aggregation. It is used for determination of size of the metallic nanoparticles or quantum dots. It is used for the quantifying the self-assembly process of polypeptides or the DNA. DLS can be used to assess the colloidal stability as a precursor to the aggregation and precipitation, and it measures the viscosity of concentrated biotherapeutics just in a few microliter of the solution. Now we will talk about the advantages of the dynamic light scattering. This technique is the accurate, reliable and repeatable on the particle size analysis. In 1 or 2 minutes, it takes a very short duration of the time. It measurements in the native environmental of the material, and mean size only requires the knowledge of viscosity of the liquid. Sample or no sample preparation, high concentration, turbid sample can be measured directly. Simple setup and fully automated measurement of the size, that is the size of more than 1 nanometer can be done, and size measurement of the molecule with the less than 1000 Dalton. Low volume is required, such as very up to 2 microliter can be used, and non-invasive measurements requires only a small quantity of the sample. Apart from this, it is good for detecting trace amount of aggregates, and it is a good technique to the macromolecular sizing. Now we will discuss about some limitation of the dynamic light scattering. The DLS is based on the scattering of light just basically measured at a very short time interval. The same limitation is for the static light scattering applies. The volume of the sample has to be higher than with some other methods depending on the specific setup, but typically 1 microliter, 1 ml is required. The concentration of the sample has to be high enough. The lower the molecular weight, higher the necessity for the concentration. The sample solution must be completely transparent, means non-turbid solution can only be employed for the DLS studies. The sample solution should not absorb light of the wavelength used, and the molecule in solution should have a refractive index that is different from that of the solvent. Apart from that, the solution should be thermodynamically stable on the same time scale of the measurement, that is, no fast reaction should take place during the solution during the measurements. So guys, let us summarize this technique, DLS technique. It is used to estimate the particle size determination by measuring the random changes in the intensity of light scattering from a suspension or the solution. This technique is commonly known as dynamic light scattering. A typical application of dynamic light scattering are characterization of the particle, emulsion or molecules which have been dispersed or dissolved in a liquid solution. The Brownian motion of the particles or molecules in suspension causes laser light to get scattered at different intensities, and analysis of these intensity fluctuation yields the velocity of the Brownian motion and hence the particle size using the Stokes-Einstein equation or relationship. In general, this technique is best used for submicron particles and can be used to measure particle size less than 1 nanometer. Now we will provide a comparison of DLS with the SLS. The output from the DLS software is often less reliable than it seems at first sight, and the results lend themselves easily over interpretation. SLS uses measurement of mean cube intensity of the scattered light at a number of different scattering angle. However, DLS measures diffusion coefficient of the particle, a measure of how fast they move in the solution. If you compare SLS with the DLS, you can see molecular weight should be calculated by SLS but not DLS. Radius of gyration should be calculated by SLS but not DLS. Second virial coefficient is also measured by SLS but not DLS. Similarly, the shape is also measured by SLS. However, the hydrodynamic radii can be measured by the DLS, not SLS. On the other hand, we should need to know the information in advance. The following information is necessary to measure the SLS. These are the weight concentration, that is in the form of gram per liter, and the refractive index increment dN by dC is required. For the DLS, we must know the viscosity of the medium and the temperature. Slide number 29 further summarize the DLS technique in the pictorial form. You can see here how the data are collected and how this instrument is work. So it's simply measured frequency intensity distribution in terms of power spectrum. The power spectrum takes from the Lorentz distribution whose half value width can be expressed at d2 dq square. All parameters in half width are known or measured. The diffusion coefficient is related to the particle size, which is given by the Stokes-Einstein equation, that is RH equals to kT by 6 pi eta d. So I hope you have understand DLS technique well. Thank you very much.